Hi, good morning. Welcome back to New Age Cosmetic White Ethic Healthcare Consultancy Daily Lectures. I'm Dr. Bobby, and I welcome all of those exam takers for the NEET Biology, NEET exams, all of the Institute of Medical Sciences, and the Council for Scientific and Industrial Research that conducts the National Eligibility Test, and for all other exam takers who are involved in biology, which is zoology, botany, and chemistry as well. Today we are dealing with the topic of molecular biotechnology, of which we're on the chapter of molecular diagnostics. We're specifically on the enzyme luciferase. So luciferase, the luciferase enzyme which catalyzes a light-emitting reaction, may be produced by a variety of different organisms including bacteria, algae, fungi, jellyfish, insects, shrimp, and squid. Though I've covered this topic yesterday, I'm just repeating the fag of the portions so that you get a continuity into your daily lectures. Please make notes, screenshots, and prepare your self-made research into the subject so you get to know more. In either case, if you have any doubts or queries, you may please post your comments on the comment section of YouTube, or you could always mail me on my email, newagecosmeticwhite at gmail, dentalrehab@gmail.com. I repeat, newagecosmeticwhite, dentalrehab@gmail.com, or you could always get back to me on my WhatsApp page at the same number, 9188661251. For those of you preparing on the exams, do make the best use of these notes. However, I request that all of you and anybody and everybody who may be accessing my lectures live, please do not commercialize any aspect of these lectures for any purpose whatsoever. We wish to thank the authors of this book, Jack, Cheryl, Glick, Bernard, Jack, and Cheryl, and all of the contributors to the various illustrations, courtesy to all of them. And we proceed to the subject matter proper. So the luciferase enzyme, as I just mentioned, catalyzes a light-emitting reaction may be produced by a variety of different organisms, including bacteria, algae, fungi, jellyfish, insects, shrimp, and squid. Luciferase genes from bacteria are termed Lux genes, while those from other organisms, the most widely studied and utilized being the firefly, are termed the Lux genes. Now, the Lux system includes 5 genes, Lux, C, D, A, B, and E, and produces a peak of light at 490 nanometers. In some applications, all 5 Lux genes are utilized as a means of monitoring the presence, the concentrations of various compounds in the environment, such as organic compounds, including phenol, salicylate, benzene, trichloroethylene, ammonia, xylene, toluene, and ethylbenzene are metals, including cobalt, copper, iron, lead, mercury, nickel, and zinc. When all 5 genes are used, the light-generating system does not require the addition of any other compounds. Therefore, following the addition of a contaminant containing sample to bacterial cells carrying Lux, C, D, A, B, and E, a quantity amounts of bioluminescence is produced within a period of a few minutes to no more than a few hours. In some cases, the reporter system includes only the Lux AB genes, while Lux A and Lux B are together responsible for generating the light signal. This system requires that the substrate decanal be added. During the assay procedure, the firefly luciferase catalyzed reactions results in the production of light at 550 to 575 nanometers. Moreover, the system requires the addition of the low molecular weight organic compound luciferin as a substrate for the light reaction. Typically, Lux genes are used in conjunction with eukaryotic cells, microbial sensors. There is a need for methods that can easily and rapidly detect the large numbers of potential toxic compounds that contaminate the environment. Once the contaminated sites have been identified and the range has been delineated, there are a number of highly sophisticated analytical techniques available to identify and quantify specific pollutants. Bacteria that are constitutively bioluminescent, that is, unlike the situation mentioned above, the bioluminescence does not need to be induced, are good candidates for pollutant detectors. In the presence of pollutants, the bioluminescence decreases, providing a clear indication of the presence of the pollutants. Naturally bioluminescent bacteria such as the marine bacterium, Vibrio fischeri, require saline conditions and a particular pH range and are therefore not useful for testing terrestrial groundwater. However, structural genes encoding enzymes that lead to bioluminescence like CDABE may be inserted into random sites in the chromosomal DNA of a soil bacterium such as Pseudomonas fluorescens. These genes do not contain a transcriptional promoter, so after insertion into the chromosomal DNA of P. fluorescens, the only luminescent colonies visualized in a darkroom are those in which the Lux genes are inserted downstream from a constituted P. fluorescens promoter. Without disrupting any important bacterial genes, the cells that luminesce to the greatest extent and have a growth rate similar to that of the wild-type strain are selected for testing with various environmental pollutants. To screen water samples for the presence of various pollutants, both metals and organic compounds, a suspension of bioluminescent P. fluorescens is mixed, with the solution being tested, and after a 15-minute incubation together, the luminescence of the suspension is measured in a luminometer. I've repeated all this yesterday. I've mentioned this, but take it for the continuity part of it, and then we go into the main. I'll just mention main points, and we go to the fresh topic. Now, in the US, before that, since this procedure is rapid, simple, and inexpensive, it is a good first screen for assessing the presence of pollution at a particular site. After a positive response with a bacterial biosensor, the actual pollutants can be determined by other methods. So in the United States, it's been estimated that there are approximately 87,000 different chemical compounds that need to be tested for estrogenic activity. That is steroid-like activity that can disrupt the endocrine system in vertebrates. While a number of different methods exist that could be used to test these compounds, they are too slow for this sort of large-scale screening. Now, at the same time that some groups of scientists are working to develop and perfect cells as biosensors, others have focused their effort on automating these systems. Such an automated system might include genetically engineered cells that emit blue-green light at about 490 nanometers in response to specific. Now, this is a diagram of the same. Take a screenshot and make notes of chromosomal DNA, wild-type bacterium, no bioluminescence, Lux genes in the chromosomal DNA, plus pollutants, and now, the compounds, an environment that sustains the cells, a light-tight enclosure, and an integrated circuit for light detection and signal processing. While simple prototypes have been constructed, the difference associated with utilizing living cells remain major impediments. And before I could proceed into the next subject of nucleic acid diagnostic systems, just a short mention that each chapter would have a summary and review questions posted as a separate video. So please see that so you don't get to forego such things. You get to see the summary, read the review questions and answer them, so you know where you stand on that particular subject, right? All right, continuing with the subject. Nucleic acid diagnostic systems. The genetic material of an organism contains the essential information that contributes to its various features and characteristics. For example, bacterial pathogenicity may be due to the presence of a specific gene or set of genes. Similarly, alteration of a gene may cause an inherited genetic disease in humans. In theory, the sequence of nucleotides that contributes to a particular biological characteristic is a distinctive signature that, if detectable, can be used as a definitive diagnostic determinant. Nucleic acid hybridization is the basis for rapid and reliable assays. The physical basis of these systems is precise nucleotide base pairing. And nucleic acid hybridization is the basis for rapid and reliable assays. The physical basis of these systems is precise nucleotide base pairing and hydrogen bonding between one string of nucleotides and a complementary nucleotide sequence. A general laboratory nucleic acid hybridization scheme is as follows. Bind single-stranded DNA, the target, to a membrane support. Add single-stranded labeled DNA, the probe, under appropriate conditions of temperature and ionic strength to promote base pairing between the probe and the target DNAs. Wash the support to remove excess bond-bound labeled probe DNA. detect the hybrid sequences that form between probe and target DNA, right? This is the picture, illustration. Please take a screenshot and make notes. The schematic representation of a strain of the yeast Saccharomyces cerevisiae has been engineered to detect low levels of estrogenic substances in the environment. Each yeast still contains the human estrogen receptor HER gene integrated into its chromosomal DNA. One plasmid BUTK4 that contains constitutively expressed bacterial LUX-CD genes and the flavinoxy reductase FRP gene from the bacterium Vibrio harvey. One plasmid BUTK47 that contains LUXI and LUXP genes under the regulatory control of estrogen response elements ERE and constitutive bacterial promoters not shown. Following the binding of the HER protein to an estrogenic compound, the complex binds to an ERE and active LUX-AB transcription. Expression of the LUX-AB, LUX-CDE, and FRP genes leads to the production of a measurable luminescence signal adapted from Sanseverino et al. Courtesy to them and thank you. Now, a nucleic acid hybridization diagnostic test has three critical elements: probe DNA, target DNA, and signal detection. This type of detection system can be both extremely specific and highly sensitive. Hybridization probes. To be effective, a nucleic acid hybridization probe must have a degree of specificity. In other words, the probe must hybridize exclusively to the selected target nucleic acid sequence. False positive, that is, responses in the absence of the target sequence, and false negatives, that is, no response when the target is present, severely undermine the utility of a diagnostic procedure. Probes can be specific at different organismic levels. They can distinguish between two or more species, determine particular strains within a given species, or identify differences between genes. Depending on the requirements of the test protocol, probes can be DNA or RNA, long, greater than 100 nucleotides, or short, lesser than 50 nucleotides, and chemically synthesized, cloned intact genes or isolated regions of a gene. Sequences that might make effective probes can be isolated in a number of ways. For example, the DNA from a pathogenic organism can be cut with a restriction endonuclease and cloned into a plasmid vector. And those plasmids that contain sequences that hybridize only to the pathogenic strain form the basis for species-specific and even strain-specific probes. Additional hybridization tests with DNA from a wide range of organisms are then conducted to ensure that the candidate probe sequences do not cross-hybridize. Each potential probe is also tested under simulated sample conditions, including the presence of mixed cultures, to determine its level of sensitivity. Wrote on the